Hey everyone, and welcome to today's explainer covering Grade 9 Biology, Unit 3, Lesson 4, Complex Patterns of Inheritance in Chromosomes in Human Heredity. I am so pumped to guide you through this today. We're going to break down some truly fascinating concepts that go way beyond the basics of biology, and will clearly map out exactly how life operates at a microscopic level.
Okay, let's run through our agenda real quick so you know what to expect. First, we'll hit our explainer objective, then move right into chromosomes and complex inheritance. After that, we'll analyze epistasis in mice, map out the central dogma, do a quick cell division misconception check, and finally, wrap things up with a formative check and recap.
Section 1. Explainer Objective. Setting our biological baseline.
So, what are we actually doing today? Our goals are to unpack chromosome linkage and recombination, trace the exact molecular flow of genetic data from DNA all the way over to proteins, and clear up a pretty massive misconception about how chromosomes actually behave when a cell divides. By the time we finish this explainer, you'll have a really solid visual understanding of all three of these pillars.
Moving right along to Section 2, Chromosomes and Complex Inheritance, going beyond Mendel.
You know Mendel's law of independent assortment, right? It essentially says that genes don't mess with each other when they're sorting into gametes. Well, here's the catch. That's really only true for genes that are on completely separate chromosomes. You see, a single chromosome is packed with hundreds or even thousands of genes, all lined up linearly, kind of like beads on a string. When genes are sitting physically close to each other on that exact same chromosome, we call them linked. They're basically a package deal, and they are far more likely to be inherited together, totally breaking that law of independent assortment.
But wait, there's a plot twist. Even those tightly linked genes can get shuffled around. How? Through a really cool process called recombination, or crossover. Right before the very first division of meiosis kicks off, homologous chromosomes duplicate and pair up. The matching genes on these homologs align perfectly with each other, and they literally swap linear segments of their genetic material. Because they're aligned so nicely, the actual order of the genes doesn't change, but the maternal and paternal alleles get completely shuffled onto the same chromosome. This creates brand new genetic combinations, meaning sometimes even linked genes can act like they're completely unlinked.
Section 3. Analyzing epistasis in mice when genes mask each other.
Out in the real world, single traits are almost always controlled by a whole team of genes working together in unison. Usually they cooperate, but sometimes they completely clash. And that brings us to epistasis. In epistasis, the relationship between genes is totally antagonistic. One gene literally overrides or interferes with the expression of a completely separate gene. We call the bully doing the masking the epistatic gene, and the one getting silenced is the hypostatic gene. It's basically a genetic veto.
Let's put this into perspective with mouse coat colors. So the normal wild-type coat color is this modeled pattern we call agouti, and that happens to be dominant to a solid-colored coat. We're going to call this the A gene. However, there was a totally separate gene, the C gene, that actually dictates whether any pigment is produced in the first place. If a mouse inherits two recessive Cs, so a CC genotype, at this separate location, it is completely unable to produce pigment. The result? You get an albino mouse, and it doesn't matter at all what that A gene is trying to do. The C gene is epistatic to the A gene.
What's really fascinating here is how this messes with our expected math. Normally, if you cross two heterozygotes for two non-interacting genes, you get that classic Mendelian 9:3:3:1 ratio. But here, because that CC allele totally masks the other gene, those two recessive groups at the end get lumped together. They both result in the albino masking effect. So our ratio actually shifts to 9:3:4. 9 agouti, 3 solid color, and 4 albino offspring. Pro tip, whenever you see a phenotypic ratio that adds up to 16 parts like this, you're almost definitely looking at a two-gene interaction.
All right, Section 4, the central dogma, the flow of molecular information.
The central dogma of biology is essentially the ultimate rulebook for how information flows in an organism. It outlines a super strict one-way street from DNA to RNA and then to proteins. Step 1 is transcription. An enzyme called RNA polymerase cruises along the DNA and builds a matching strand of messenger RNA. Step 2 is translation. This mRNA heads out of the nucleus, finds a ribosome, and the ribosome basically reads the genetic code in chunks of 3 bases to string together a line of amino acids. Finally, step 3. That long chain of amino acids folds up into a working, functional protein. And that protein is what actually goes off to do the heavy lifting for the organism.
Now, I said it was a one-way street, from DNA to RNA to protein, but biology always has exceptions, right? There are absolutely special cases that break the rules. For example, some RNA can literally copy itself using an enzyme called RNA-dependent RNA polymerase. And then there's reverse transcription, which is just wild. Retroviruses carry this enzyme called reverse transcriptase that takes their RNA genome and literally writes it backward into the DNA of their host cell. Even our own healthy cells do a version of this. An enzyme called telomerase uses a tiny piece of RNA as a template to build DNA onto the ends of our chromosomes, which stops them from getting too short every time our cells divide.
Section 5. Cell division misconception check. Correcting the visual model.
Before we wrap up, we absolutely have to address a massive misconception students often have about how chromosomes behave during cell division. It's super common for people to assume that the actual number of chromosomes magically doubles during prophase of mitosis and then gets cut in half during anaphase. This confusion usually pops up because it is really easy to mix up the concept of a chromosome with the concept of a chromatid.
So, let's set the record straight. During interphase, the DNA material itself definitely replicates to form two identical sister chromatids for every single chromosome. But, and this is absolutely crucial, the actual counted number of chromosomes in that cell does not double during prophase. A replicated chromosome, which is made of those two sister chromatids joined right at the centromere, still just counts as one single chromosome. Yes, the amount of genetic material has doubled, but the actual chromosome count stays exactly the same until those chromatids get physically ripped apart later on in the process.
Section 6. Formative check and recap. Testing your knowledge. Let's see if you can put this all together. Think back to our mouse coat color example for a second. Why exactly did that genetic cross fail to produce a standard Mendelian dihybrid ratio? Why did we get 9:3:4 instead of 9:3:3:1? Take a quick mental pause and try to recall the specific mechanism we talked about.
If you guessed epistasis, you hit the nail on the head. The C gene is epistatic. That recessive CC genotype totally blocks any pigment from being produced. Because it completely masks whatever the A locus is trying to do, it collapses those final two expected categories from the 9:3:3:1 ratio into just one single combined group of 4 albino offspring.
Let's do a super quick recap of the required concepts we hit today. We explored how linked genes on the same chromosome basically travel together, unless they get separated by the shuffling of recombination. We modeled epistatic interactions where one gene completely overrides another. We traced the central dogma straight from DNA to RNA to protein. And finally, we cleared up a major misconception, confirming that chromosome counts definitely do not double during prophase.
I'm going to leave you with this final thought to chew on. We just looked at some incredibly strict microscopic processes, from masking genes to reverse transcription to chromosomal crossover. But how do all these rigid molecular rules and complex inheritance patterns somehow work together so seamlessly to build the insanely unpredictable and vast biological diversity we see every single day on our planet? It's a massive puzzle, and it's absolutely fascinating. Thank you so much for joining me for this explainer, and keep exploring.